Imagine a world without genetic diseases. Imagine crops engineered to resist pests and thrive in challenging climates. This is the promise of CRISPR, a revolutionary gene editing technology that has taken the world by storm. CRISPR allows scientists to precisely target and modify DNA sequences, offering unprecedented control over the building blocks of life. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. This mouthful of a name refers to a unique feature found in the DNA of bacteria. These repeats, it turns out, are part of an ancient immune system that bacteria use to defend themselves against viruses. Scientists have harnessed this bacterial defense system to create a powerful tool for gene editing. The potential applications of CRISPR are vast and wide-ranging. From treating genetic diseases to developing new agricultural products, CRISPR is poised to revolutionize many aspects of our lives. However, this groundbreaking technology also raises profound ethical and societal questions. As we stand on the cusp of this genetic revolution, it is crucial to engage in thoughtful dialogue about the potential benefits and risks of CRISPR. We must proceed with caution and responsibility, ensuring that this powerful tool is used for the betterment of humanity and our planet. At its core, CRISPR is a molecular scissors that can cut DNA at specific locations. This precision cutting is made possible by a protein called Cas9, which acts as the blade of the scissors. Cas9 is guided to its target DNA sequence by a short RNA molecule called the guide RNA. The guide RNA is like a GPS navigation system, directing Cas9 to the precise location in the genome that needs to be edited. Once Cas9 reaches its target, it makes a double-stranded break in the DNA. This break triggers the cell's natural DNA repair machinery, which can be harnessed to introduce specific changes into the genetic code. There are two main ways that CRISPR can be used to edit genes. In the first method, CRISPR is used to disrupt a gene by introducing a small insertion or deletion. This disrupts the gene's function, effectively turning it off. In the second method, CRISPR is used to introduce a new piece of DNA into the genome at the site of the cut. This allows scientists to replace a faulty gene with a healthy copy, or to introduce a new gene altogether. The simplicity and versatility of the CRISPR system have made it a game-changer in the field of gene editing. It is faster, cheaper, and more accurate than previous gene editing technologies, opening up exciting new possibilities for research and applications. CRISPR is already making its mark in various fields, offering innovative solutions to some of our most pressing challenges. In medicine, CRISPR holds immense promise for treating genetic diseases. Scientists are exploring its use in correcting genetic defects that cause diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease. Clinical trials are underway to test the safety and efficacy of CRISPR-based therapies for various genetic disorders. For example, researchers are investigating the use of CRISPR to treat beta thalassemia, a blood disorder that reduces the production of hemoglobin. By correcting the genetic mutation responsible for the disease, CRISPR could potentially offer a cure for this debilitating condition. Beyond treating genetic diseases, CRISPR is also being explored for its potential in combating infectious diseases. Scientists are developing CRISPR-based strategies to target and destroy viruses like HIV and hepatitis C. By disrupting the viral genome, CRISPR could prevent these viruses from replicating and spreading. In agriculture, CRISPR is being used to develop crops that are more resistant to pests, diseases, and harsh environmental conditions. This could lead to increased crop yields, reduced reliance on pesticides, and enhanced food security. For example, researchers have used CRISPR to create tomatoes with increased shelf life and rice varieties that are resistant to bacterial blight. While the therapeutic applications of CRISPR are undeniably promising, the possibility of using this technology to enhance human capabilities raises profound ethical concerns. Imagine a future where parents could choose their children's physical traits, cognitive abilities, and even personalities. This prospect, once confined to the realm of science fiction, is now within the realm of possibility thanks to CRISPR. Proponents of human enhancement argue that it could lead to a healthier, more intelligent, and more capable human race. They envision a future where genetic diseases are eradicated and human potential is maximized. However, critics warn of the potential for exacerbating existing social inequalities and creating a genetic divide between the enhanced and the unenhanced. The ethical implications of human enhancement are complex and far-reaching. Who gets to decide which traits are desirable and which are not? What are the potential consequences of tampering with the human germline, potentially affecting future generations? These are questions that demand careful consideration and societal debate. Section 5. The Ethical Maze, Navigating the CRISPR Revolution. The rapid pace of CRISPR research has outpaced our ability to fully grapple with its ethical implications. The potential to alter the human germline, the DNA that is passed down from one generation to the next, is particularly concerning. Changes made to the germline would be inherited by future generations, raising the specter of unintended consequences. The possibility of designer babies, where parents could select desirable traits for their offspring, raises concerns about genetic discrimination and the commodification of human life. It also challenges our understanding of what it means to be human and the value we place on diversity and individual uniqueness. Moreover, the equitable distribution of CRISPR technology and its potential benefits is a pressing concern. Will everyone have access to these transformative technologies, or will they only be available to the wealthy and privileged, further widening existing social disparities? These are just some of the ethical challenges we must confront as we navigate the CRISPR revolution. Section 6. Unforeseen consequences. Weighing the risks of CRISPR. While the potential benefits of CRISPR are undeniable, it is crucial to acknowledge and address the potential risks associated with this powerful technology. One concern is the possibility of off-target effects where CRISPR inadvertently edits genes other than the intended target. This could lead to unintended mutations with unknown consequences. Another risk is the potential for mosaicism, where CRISPR edits are not uniformly distributed throughout all cells of an organism. This could lead to unpredictable outcomes and complicate efforts to assess the safety and efficacy of CRISPR-based therapies. Furthermore, the long-term effects of CRISPR gene editing are still largely unknown. It is possible that CRISPR edits could have unintended consequences that only become apparent after many years or even generations. Careful monitoring and long-term studies will be essential to assess the safety and stability of CRISPR-modified organisms. Section 7. CRISPR and the future of human evolution, a paradigm shift. The advent of CRISPR has profound implications for the future of human evolution. For millennia, humans have been shaped by the forces of natural selection. However, CRISPR gives us the power to directly manipulate our own genetic destiny. This raises fundamental questions about what it means to be human and the future of our species. Will we use CRISPR to enhance our physical and cognitive abilities, potentially creating a new era of transhumanism? Or will we focus on using CRISPR to address genetic diseases and improve human health and well-being? The choices we make today will have far-reaching consequences for generations to come. The CRISPR revolution is a defining moment in human history. It presents us with both unprecedented opportunities and profound ethical challenges. As we venture into this uncharted territory, we must proceed with caution, humility, and a deep sense of responsibility for the future of our species and our planet. Section 8. Balancing innovation with responsibility, a call for global dialogue. The ethical and societal implications of CRISPR are too significant to be left solely in the hands of scientists and policymakers. A broad societal dialogue is essential to ensure that this powerful technology is used responsibly and for the benefit of all humankind. We need to engage in open and transparent discussions about the potential benefits and risks of CRISPR, involving diverse voices and perspectives. This includes ethicists, philosophers, religious leaders, policymakers, and the general public. International collaboration and the establishment of clear ethical guidelines are crucial to prevent the misuse of CRISPR and ensure its equitable distribution. We must work together to harness the transformative potential of this technology while safeguarding against its potential risks. Section 9. Conclusion. Embracing CRISPR's potential while treading cautiously. CRISPR is a powerful tool with the potential to revolutionize many aspects of our lives. From treating genetic diseases to developing sustainable agriculture, the possibilities are vast and inspiring. However, we must proceed with caution, recognizing the ethical complexities and potential risks associated with this groundbreaking technology. As we stand at the forefront of the CRISPR revolution, it is our collective responsibility to ensure that this powerful tool is used wisely and for the betterment of humanity. By engaging in thoughtful dialogue, fostering international collaboration, and prioritizing ethical considerations, we can navigate the challenges and opportunities presented by CRISPR and shape a future where this transformative technology benefits all of humankind.